Hey everybody, what's up? It's Mr. Dan, and in this video we're going to introduce ourselves to DNA and what it is and how it was discovered. Okay, this will be a two-part video, so watch both of them, please. So before we do that, let's just look closely again at our standard. So what we're doing here, it says construct an explanation based on evidence for how the structure of DNA determines the structure of proteins, which carry out the essential functions of life through systems of specialized cells. So, structure of DNA changes structure of protein, which determines function of cells. Okay? Nice little chain there to follow. Let's skip through a few slides to get to where we need to go. All right, so what is DNA? That's what we want to answer in this video. And we're going to look specifically at how it was discovered. So DNA as genetic material. DNA is the genetic material that allows traits to be passed through the generations. That's what genetic means. It's from one generation to the next. Okay, so DNA, it's the thing, it's the material that does that. Every organism has billions of identical copies of DNA. Every cell in your body has a copy of DNA, and that DNA is exactly identical in every single cell. And every single organism does this. My DNA might be different from yours, but every cell in you is the same, and every cell in me is the same. It is a code for every structure and function that goes on in the body. It is the code that can be translated as a blueprint for life. So everything about you is written. Everything about you, like as a physical living thing, is written on that DNA. It can be copied and passed through the generations, and every individual has a unique code. Like I said before, mine is different from yours, but they're probably quite similar. Ours is different from a whale's, though there are similarities. There's similarities through different organisms, but everybody has a unique code, whether you are the same species or not. Okay, so we're going to go through a couple of scientists and experiments to try to show how we discovered that DNA is in fact the genetic material. And this will help us understand a bit about the nature of DNA as well. So maybe quickly, DNA, it's called deoxyribonucleic acid. Okay, so it's a group of chemicals called nucleic acids, and there are others. There's DNA, there's RNA, there may be others, I'm not so sure, but nucleic acid. So this first experiment is done by a guy named Frederick Griffin. Okay, he's this British bacteriologist, and he did an experiment with mice and a bacterial strain called pneumonia. Pseudomonas pneumoniae, I think is the official term for it there. So this is a bacteria, and it can cause disease. I think everyone who's watching this video has had pneumonia at some time. It's a respiratory infection. You end up with some fluid in your lungs. Usually when you've got the common cold and a cough, you got a bit of pneumonia. It can get bad, or it can be kind of mild. Okay, so bacteria, they often have many different strains, and this is no exception. There are two strains that we're looking at here of pneumonia. One is a smooth strain, and one is a rough strain. The smooth strain causes disease in the infected organism, and if it's a mouse, it's going to die. So the infection is strong enough to kill it. On the rough strain, when you inject it into the mouse, nothing happens. It survives. So non-virulent and virulent are the official terms here for it. This is called smooth because when you look at it under a microscope, there's a protective coating, this yellow part here, and it looks smooth. The blue ones, the rough, there's no coating, so they're called rough ones. All right, so smooth causes death, blue, mouse survives. Then they took the smooth ones here, and he put them under fire, and he killed the strain. So the bacteria itself was dead, and when he put that into the mouse lives. Okay, so there's something going on here, but the mouse died. So the clincher here is the final trial And here he took two different things that do not kill the mouse. The blue strain, the living blue strain, which does not kill the mouse, and the dead smooth strain, living rough strain, which does not kill the mouse. Dead smooth strain, which does not kill the mouse. And a combination of these two in the syringe killed the mouse. So what happened here? Well, what Griffin suggested in this experiment was that whatever it is about this smooth strain that killed the mouse, it survived this heat treatment here in killing it. And then whatever that thing is, whatever that material is, the genetic material was, it was able to be transferred into the blue, into the rough strain, and then kill the mouse. So that was what he suggested. He had no idea what that was, but he was able to say that there is a material inside of a cell that causes death that can be transferred from one organism to the next. That's genetic material. So what is that genetic material? The next guy, called Oswald Avery, here he is, he won a Nobel Prize. He did the same experiment, only this time he he was able to be a little bit more specific in doing it. Scientists at the time, they knew that DNA existed. They knew about nucleic acids. They knew about proteins, lipids, carbohydrates. They know all sorts of biochemical molecules, but they just didn't know what they all did. Most scientists were in agreement that it was either nucleic acid or it was protein. And so he did the same experiment, and then when it was time to inject the combinations here, what he did is he took this heat-killed smooth strain and he isolated out proteins from it, and he isolated out DNA from it. It's not so hard to do. It's an experiment that we'll actually do in class. We can isolate DNA from living cells, no problem. I'll show you how. So he separated out proteins and he separated out DNA, and then he injected those into the mouse. Well, guess what? The protein-injected mouse survived, and the DNA-injected mouse died. So he was able to add some confirmation to Griffin's experiment that says that it's DNA, in fact, that is the genetic material. There was still some questioning of these results at the time, though, and so it just required further investigation by some other scientists. And so Alfred Hershey, this guy, and Martha Chase, this woman right here, they did some experiments using bacteriophages and bacteria. A bacteriophage is a virus that infects bacteria only. So these are harmless to us. They're everywhere, but they're not going to hurt you at all. So they had two different experiments. They had one where they were specifically testing proteins, and then another where they were specifically testing DNA. Okay, so how they did this is that they cultured a whole bunch of bacteriophages in two different growth mediums. One for the proteins was grown in a radioactive solution with radioactive sulfur on it. Okay? And then the DNA had radioactive phosphorus on it. So why is this important? Well, scientists know that, I mean, you're going to know this in about 30 seconds because I'm going to tell you. Proteins have sulfur in them. Sulfur is an important part of all proteins, of amino acids, but there's no phosphorus. DNA has phosphorus in them, but they don't have any sulfur. And they knew that. Scientists knew this at the time. They knew about these different molecules and the chemical makeup of them. So they identified these two unique elements in each type of molecule, and they grew it up in this radioactive solution. So what that does is it allows us to track this from one organism to the next. We can see where it is. Okay. So let's maybe take a look here really quick at what a bacteriophage is, just to add on to that a bit. Viruses are generally no more than DNA and protein. Okay, so proteins are what have the sulfur, DNA is what has the phosphorus, and so if the outside part is coated in something radioactive, if that protein part is transferred from one organism to the next, we'll be able to track it. If the radioactive DNA is transferred from one organism to the next, we'll be able to track that too. I hope that makes sense what's going on here with this. All right, so they do it, they grow it up, the protein, the radioactive proteins on the viruses, the viruses inject they do their thing, they infect the bacteria, and then the bacteria, the way these work is they basically just inject their genetic material into the organism, and then that bacteria starts producing a whole army of viruses. That's how these work. So in doing so, what was observed is that in the protein injected, or in the sulfur labeled viruses, the bacteria cells that resulted from this did not have any radioactivity in those cells. In the DNA phosphorus labeled things, the radioactivity was detected in the resulting bacteria cells. So here the proteins did not get injected into the cell. Here the DNA did get injected into the cell. So in both, DNA was injected, but only the radioactive DNA was detected, and the radioactive proteins were not. And if you were to take the resulting thing that was going to happen here, what you would see if you centrifuge them down is that you would have a whole bunch of cyprinate, which is the liquid that remains after a centrifuge, and that's full of the protein shells, and then the DNA, which is more dense, would be down here. Here, the dense DNA is all radioactive, and then the liquid that includes the protein shells of the viruses is clear, meaning that it's not radioactive. So this is a very good experiment that confirms the idea that DNA is in fact the genetic material. All right. Just a little bit more on this then. Now that it was determined that in fact DNA is the genetic material, we wanted to get into the idea of what is the structure of it. Okay, we want to know the structure of DNA. The structure of DNA can lead to an understanding of the function of it. So it tells us how it works. Take a look at anything, and how it looks says a lot about what it's actually capable of doing. All right, so first thing, as far as trying to discover the structure of DNA, a guy named P.A. Levine discovered that the building blocks of DNA were these things called nucleotides. And a nucleotide has a basic structure where it's got a phosphate group, it's got a sugar, and it's got a nitrogenous base. Okay, so he discovered that, and he also discovered that there are 5 different nucleotides that exist. They fit into 2 categories called purines and pyrimidines. You can see a difference in these 2 structures here. Guanine, adenine, cytosine, and thymine, these are all part of DNA. And then there's a RNA that we'll talk a lot about as well. Instead of thymine, they have uracil instead. So a subtle change, but more on that later. Here's a more detailed look at what one of those nucleotides might look like. Here's what a phosphate is. It's phosphate with sugars. Phosphate with oxygens. Sugar is a carbon ring with some oxygens and hydrogens on it, and then the nitrogenous base is it's mostly nitrogen and carbon. So we knew that. Thanks to P.A. Levine, we knew what the building blocks of DNA are. And then the next scientist of note is a guy named Erwin Chargaff. And we have this thing called Chargaff's rule. And what this tells us is that in any organism, the the percent by composition, the amount of adenine, right, of this one, it's equal to the amount of thymine that's present. And cytosine and guanine also come in equal ratios. So this is a really important thing. We can try to draw some conclusions, and maybe you know what this means, but this is his contribution, that the percent of A's and T's are equal, and the percent of C's and G's are equal. So that's a valuable piece of information that we'll use later as we go. Okay, so this is the end of part 1, and then I'm going to make part 2, and you guys can watch that also. All right? So thanks. Hope this is helpful for you. See you soon.